Biology is full of variables and uncertainty. Biostatistics has helped us make sense of it all. But what if our math tools are missing some important clues? Now, let's look at the three biggest challenges in biostatistics that still puzzle even the smartest scientists. Let's start with a big genetic mystery: the missing heritability. After the Human Genome Project, scientists thought we finally had the instruction manual for humans. Researchers began conducting large genome-wide association studies, or sometimes known as GWAS, to link specific DNA variants to disease, such as diabetes and schizophrenia. We use statistics to estimate inherited traits, but even when we account for genetics, there is still a big gap. Take height, for example. Traditional pedigree and twin studies estimate that about 80% of height difference are genetic. But when researchers use genome-wide association studies to identify the exact single nucleotide polymorphisms, or SNPs, responsible, the genetic markers they find explain only a small part of the variation. So what is missing? Are those genes just hidden from us? For years, early genome-wide association studies focused on common variants, which are mutations found in more than 1% of people. But now we know that much of the genetic variation comes from rare variants. These are like genetic typos that only shows up in a tiny number of people. Because they are so rare, regular statistical model cannot easily link them to a trait. The signal gets lost in those noise. Also, many traits are not controlled by just one or two super genes. Instead, thousands of variants each have a tiny effect. This is where biostatistics becomes really complex. Traditional genome-wide association studies models usually look for additive effects, assuming that gene A plus gene B gives the final trait. But biology is rarely this simple. Sometimes gene A only causes a disease when it interacts with gene B. This is called epistasis, or gene-by-gene interaction. There is also gene-by-environment interaction, in which genetic risk, such as those for obesity, can manifest in one environment but remain hidden in another. There is another gap in heritable information that is not directly written in DNA. Epigenetics, which involves chemical change that turn genes on or off, is shaped by factors like environment, stress, and diet. And I know this is a bit of an oversimplification of epigenetics definition, but it essentially means that figuring out how these environmental factors are passed down either alongside or independently of the genetic code remains a challenge for today's models. Researchers are still working to solve this puzzle. Our second challenge is the causal inference problem, and we know that the science golden rule is that correlation is not causation, but biostatistics is still trying to figure out what really causes what. Think about this. Data might show that people who take a certain vitamin supplement live longer, but is it the vitamin that helps them live longer, or is it just health conscious people are more likely to exercise, eat well, and buy vitamins? biostatisticians have developed advanced mathematical tools, such as directed acyclic graphs and propensity score matching to infer cause and effect without needing a perfect clinical trial. But these models rely heavily on assumptions. If a scientist is wrong about even one variable, the whole model can fall apart. The real challenge is that we can measure things very precisely, but proving exactly why and how something happened is still a big debate. Our next challenge strikes at the science core, the replication crisis. In biostatistics, a p-value below 0.05 is the gold standard, suggesting findings are not flukes. It means less than a 5% likelihood the results is pure chance. Still, a p-value of 0.049 gets headlines and attention, even though a small change could tip the results. Small tweaks like changing criteria or excluding outlier can produce the outcome people want. This is called p-hacking. It makes people trust biology less. When studies are repeated, the results often change. The replication crisis makes us ask, how can we protect research if statistics can be changed so easily? Furthermore, should a p-value below 0.05 be the golden standard? Or is it even acceptable in medical statistics? Would you accept a 5% treatment failure just because it occurs by chance? This challenge reminds us that science is not about knowing everything. It is about understanding what we do not know. So, when you hear news about disease, genes, or miracle drugs, remember that biostatisticians are still working hard to solve the math behind life. And that is it for this video. Thanks for watching.